Hello everyone, I am Dr. Hemlata Srivastav, working as Assistant Professor in School of Biosciences in IMS College, Ghaziabad. Here I am going to explain you history and introduction of bioinformatics. So first of all, I am going to explain you about the bioinformatics. Bioinformatics is a novel discipline that need to manage, interpret the genomic data that was massively generated by genomic research from past decades. This discipline opened different new avenues in the field of biology. This connect to the genomics to the information technology also. It also connect different field of the science. It is statistical analysis, which helping to interpret data, mathematics, and which is helping to develop a mathematical model in biological phenomena, and it also helping develop new algorithm, which is helping to develop new tools and techniques in the field of biology. So for this require different skills in a bioinformatics field. It is biology, computer science, mathematics or statistics, or information technology. But all these skills are not present in a single person. That's why we require a team in which present a programmer, biologist, IT person, statistician, and graphic designer. All the person work together and work on bioinformatics. So bioinformatics is a process which connect genotype to phenotype. Actually, it is the genetic data, and phenotype is the expression study which is visible in the environment. It is visible as a physiological disease, as a particular expression, as a particular characteristics which is properly visible in an organism. So this physiological expression understand by the genetic study. And in the genetic study, there are molecules, DNA, RNA, proteins, which are connect together and develop a molecular networks which are expressed in a particular cells and this expression would be positive, would be or negative. If it would be negative, then it would be comes as a physiological disease. So for this study the physiological disease, we require understand the genomics. It is DNA in an ACTG format. It would be sequence, and for this we need to understand the sequence analysis, and we need to do sequence alignment for comparison the sequence from the normal person to the diseased person. Through this similarity study, we can identify the new mutation which is responsible for this disease, and we can identify a new gene which is responsible for the disease or a particular function. Through this, we can compare the normal person through the diseased person and understand the evolutionary pattern of a particular mutation or particular the DNA structure or particular molecule such as protein. Through this understanding, we can understand the protein network who are expressed in a cell and responsible for the disease. So here we can understand how the genotype convert into the phenotype and how the bioinformatics helping to understand whole genetic information and how this genetic information is helping to understand the phenotype of in a particular person. So when we connect this information, when we collect this information as a data, all the sequence information in ACTG format, it needs some management. So first we need to manage whole data as a databases. Then we need to study rigorous analysis in that data and using the computer algorithm and develop new algorithm or software which is helping to understand this data, genomic data, then develop new discoveries in biological sequences, and further analyze and do the simulation study and predict new analysis and predict new discoveries in the whole biology, and which is a beautiful study of the bioinformatics. So what we do in the bioinformatics, first of all we do the sequencing. That means we convert our data into ACTG format, and then we have to assemble that sequences. Then we compare the sequence and understand the relationship, which is called phylogenetic analysis. And after this relationship and assembly study, we can understand or predict a particular new gene, new mutation, or new protein which is responsible for a particular behavior or particular expression. Further, we have to annotate our sequence into the exon and intron, and then we have store whole data in a database which and share to the other persons, to the other scientists. So, how this bioinformatics word or terminology developed? It is developed in 1970 by the Hesper B and Hujwit. These are the two scientists who has taken this, who has used this term bioinformatics as a bioinformatica in their first paper in 1970. Next scientist is the Margaret Oakley Dayhoff. In the duration of 1925 to 1983, she is a chemist who has developed a new computational method in the electrochemistry study, and then worked together with the Robert S. Ledley for the solving the problems in the biomedical sciences. Both are them together worked and developed a new program, computer program. It is ComProtein, and designed and developed a novel program also. It is IBM 7090. Both the programs work together to determine a new or primary protein structure. After that, they have developed a first software in the biological sciences is Fortran for assembly the sequences. So this is the ComProtein, which is developed by the Dayhoff. Dayhoff is the first bioinformatician. She is the father and mother of the bioinformatics because she has started actually bioinformatics in the biological sciences from 1970 or in the period of 1962. They have developed this program for understand the protein structure. After that, she has developed a new database which is based on the protein sequences. This is the first database, Atlas, which is first ever biological sequence database. It has 65 protein sequences. These protein sequences datasets reflect the evolutionary history of the species. So Atlas developed in the 1965 by the Dayhoff, and this is very beneficial information in the biological sciences. So outcome of the study: to determine the sequence pattern of the organism, to identify the sequence similarities and dissimilarities between organisms through the bioinformatics. Through the bioinformatics, we can understand the structural variation of the proteins in different organisms. We can understand or define the complexity in three-dimensional structure of the protein and nucleic acid, it is DNA or RNA molecule. We can understand the variation in functional behavior of the different organism. We can understand also the phylogenetic relationship to identify the ancestral species of a particular sequence.